All right, today's lecture is going to be on Chapter 10, which is Precipitation and Agglutination Reactions. This lecture is only about 20-some slides long, so we're going to do it in one session rather than breaking it up into several sessions. All right, some objectives here. The first thing we're going to start with is talking about antigen and antibody affinity and avidity. We've spent some time talking about the different parts of an immunoglobulin. We talked about the Fab region, which is the area that binds onto the antigen or the epitope, and the FC region, which is more of the stalk, which looks like the tree trunk of the antibody. So these two definitions you're going to have to be familiar with. When it comes to antigen-antibody binding, first we look at the affinity. The affinity is the initial attraction force between the Fab site on an antibody molecule and the epitope or determinant site on the antigen. So that is essentially where those Fab hands that I always mention in class grab onto that antigen. So what is the attraction? How well do they fit together? The strength of the attraction depends on the specificity of the antibody for a particular antigen. So the more they fit together like a lock and key, the better the affinity will be. So they kind of need to match. The more the cross-reacting antigen resembles the original antigen, the stronger the bond will be. So you can see in the picture on the side here, if they look more similar, they're going to have a stronger affinity for each other. The next term is avidity. This is the sum of the attractive forces between an antigen and the antibody that keeps the molecules together. So I think of somebody who would be like an avid exerciser, would be very strong, right? So that would be the strength which holds the antigen and the antibody together. Affinity is how well they fit together. Avidity is the strength of the fit when they come together. So there is a law of massive action. I'm not going to make you do any calculations like this on your test, but there is kind of a way that these fit together. So the free reactants are in equilibrium with a bound reactant. You can see antigen-antibody binding divided by the antibody concentration. The antigen concentration is what's in this calculation above. And the value of K depends on the strength of the binding between the antibody and the antigen. The higher the value of K, which is that strength, the larger the amount of antigen-antibody complex that we'll see, and the easier it is for us as laboratorians to be able to detect the reaction. So we're going to talk about precipitation and agglutination reactions along with a few other ones as well, but we're going to start with precipitation. When I think of precipitation, I think of like when it rains outside. So this involves combining a soluble antigen with a soluble antibody. If something is soluble, that means that it's able to dissolve in a liquid. That's how I think of it. So when you add your Kool-Aid powder to water, it kind of dissolves. Kool-Aid powder would be kind of soluble. Once it's in the liquid, it kind of just stays. It doesn't precipitate out or anything. It just stays liquid within there. So when we take a soluble antigen and a soluble antibody and they combine together, we have a visible reaction called precipitation. So we have to have multiple binding sites for each other, and we have to have equal relative concentrations of each. One example of this is the flocculation test. There's a picture down here of what flocculation might look like. So in the non-reactive first well that you see down in the bottom, this one doesn't have any agglutination or clumping, okay, or precipitation. As we move over, we've got weak. This one's difficult to see, but our reactive ones have clumping that we can actually visualize. So that's what we're looking for a positive test. This means that the antibody combined with the antigen and created that look. There's a couple definitions that you're going to be familiar with as well, which is the zone of equivalence along with prozone and postzone. The number of multivalent sites of an antigen and antibody should be approximately equal. This is where we hit the zone of equivalence. You can see in the middle of this picture here that we've got antigens and antibodies in equal numbers. As we move to postzone, this is where we have excess antigen. There's so much that you almost can't see the reaction because there's way too much antigen present. On the other side, we have prozone, where there's way too many antibodies where we may not be able to see a reaction. Ideally, we want to be in the zone of equivalence, which is where we will see the best precipitation or agglutination happening. So here's a little more information on each one. So the prozone phenomenon, which is antibody excess, is when antigen combines with only one or two antibody molecules. My students always remember this one by saying that pro football players have good bodies. So prozone and antibody excess go together because you're going to have to know that postzone is antigen. But prozone, pro football players have good bodies, antibody. There's no cross linkages are formed. We get a lot of false negative reactions as a result of having way too much antibody. And if a false negative reaction is suspected, we dilute out the antibody and perform the test again. On the other side of the spectrum, we have that post zone. That's where there's too much antigen. We have small aggregates that are surrounded by excess antigen. We don't get a lattice network formed, and we have the presence of a small amount of antibody that can be obscured, giving us false negative results once again. And in this case, the test should be repeated to allow further antibody production, or if the test is negative again, it's unlikely that the patient actually has the antibody. So here's some definitions of a couple different types of precipitation techniques that are used. Nephelometry is something that we've already mentioned. This is where the light is scattered at an usually about a 90-degree angle due to the amount of turbidity that is created by the antigens and antibodies forming together. Radial immunodiffusion is taking immunoglobulins and allowing them to diffuse through a gel. And the last one here, Auchterlonie double diffusion, which we'll see a picture of here in a little bit, uses antigen and antibody that diffuses out of the well, and we look at the relationship of those antigens between each other. And we're actually going to be performing that in lab during week 7. Another one, immunoelectrophoresis. We allow antibodies to flow through a gel using electricity. And immunofixation electrophoresis, which is where we can find the over or under production of an antibody. So that's electrophoresis as well, but we apply the antibodies to the top of the gel. So these are things that we'll see some more information about as we move through. All right, so the first one we'll talk about is radial immunodiffusion. This one may sound familiar because we talked about it with the complement. So radial immunodiffusion is a single diffusion technique where antibody is in the gel and the antigen is applied to a well cut into the gel. Antigen will diffuse until the endpoint of equivalence is reached. So those antigens and antibodies want to stick together. It's kind of like a magnet, like a magnetic attraction. So it will travel through the gel to meet an area of equivalence within that gel that we can actually measure. So the square of the diameter is proportional to the antigen concentration. So Becky, what does that look like? Here we go. So we have a couple different ways. So we have a glass slide coated with agar containing a specific antibody, or we have a gel in like a petri dish on the right hand side that you can see. So these specific items are applied to the well in the gel and allowed to diffuse outside of the gel towards their antigen or antibody to determine how much is there. And we measure that zone, this clear zone here that you see. Not really clear, I guess. It's like a hazy zone. We measure across that to determine the concentration. The next one is Auchterlonie diffusion. This one is a double diffusion technique. Wells are cut into a gel and both the antigen and antibody diffuse out radially. A line of the precipitate forms where the antigen and antibody meet. There's 3 possible patterns: identity, partial identity, or non-identity. So here's a picture of this one. Okay, you can see that the antibody is added, okay, and we have antigens in the well 1, and they will diffuse out towards their respective antigen. Depending on whether there's identity or not identity, we look at these different arcs. This one has an absolute identity. It creates an arc, okay. Non-identity, we're going to see this X pattern. Partial identity, it kind of forms a T. Another important thing down here that you see, the density of the line reflects the amount of immune complexes formed. So you can see this one down here, how dense that is. There's a lot of immune complexes inside that dense line. The next one, immunofixation electrophoresis. So this is also a double diffusion technique where unknown antigen is electrophoresed and then antibody is placed on the top of the gel. Precipitates form where the antigen-antibody combination has taken place in the gel. So this is used with serum as the antigen to determine over or under production of antibody types. So here's a picture of what that might look like. Okay, we have different types of immunoglobulins on here. We've got G, A, or IgG, IgA, IgM, and we've got some kappa and lambda down here on the end as well. All right, our next type of reaction is going to be agglutination. So this is the visible aggregation of particles resulting from the combination of a specific antibody. There's two steps you have to be familiar with. The first one is sensitization. That is the initial binding. This is where the antigen and antibody unite together through antigenic determinant sites. Then the second part is lattice formation. Once the antigen and antibody bind together, we form larger aggregates where the antigen and antibody bind to form a stable lattice. This is produced by antibodies called agglutinins. So you can see we have the antibody. On the side, we add it to antigens that have multiple determinant sites on it. You can see the little antigen sticking off the side. Sensitization, we can't see, but you can see there's antibodies actually attached. After a while, they all come together and create this pretty lattice formation. And this is what we see as agglutination. The first type of agglutination is direct agglutination. So this uses known bacterial antigens to test for the presence of unknown antibodies in the patient. The first one we'll talk about is the Weidel test. This is a rapid screening test to look for typhoid fever. It uses Salmonella O and H antigens and to look for Salmonella. Direct agglutination, hopefully this looks familiar from our lab that we did in class week 5, but this is direct hemagglutination. It involves red blood cells, and the example of this would be ABO blood group typing, which is what you're going to be doing for your virtual lab this week. So here's another example of what we did. So after we centrifuge, we had the red cell button. We shook it. If there was one solid clump, we called it positive. We called all of these positive. But in the blood bank world, we do rate those as 4+, 3+, 2+, 1+, but you can worry about that when you get to blood bank class. We also can do the same test on a slide. So we can add some antigen and antibody on a slide, mix it together, and we can see the clumping taking place on the slide as well as a tube. The next type is passive agglutination. Passive agglutination employs particles that are coated with antigens not normally found on their surfaces, such as erythrocytes, latex, gelatin, silicates. I would say the most common one that you'll come across in medical laboratory science is latex. So these are synthetic beads or particles that provide consistency, uniformity, and stability. It makes that reaction easier for us to see. Some of the tests we use this for: rheumatoid factor, group A strep, antibodies to rotavirus, CMV, rubella, varicella, hepatitis, HIV. All those tests, we use this type of reaction. So the antigen is attached to a carrier particle, usually something like latex, and agglutination occurs if the patient antibody is present. So again, some of the things that we use, this is a repeat of what you just saw, to detect rheumatoid factor and some other types of usually viruses or bacteria. Reverse passive agglutination is opposite of what you would think. So this is used to detect microbial antibodies. Some common applications for this, looking for group B strep, staphs, strep groups A and B, rotavirus, cryptococcus. We're looking for soluble antigens in urine, spinal fluid, and serum. So in this case, the antibody is attached to the carrier particle rather than the antigen. So agglutination occurs if the antigen is present in the patient sample. We do this a lot for staph bacteria in microbiology. So if we think something might be staph, we can take a latex solution that has staph antibodies attached to those latex beads. We mix it with some of what we think might be staph, and if agglutination happens, we know that is staph bacteria. So here are some pictures and some comparisons. So on the top here, we have the carrier particles mixed with a soluble antigen. So we've got maybe a latex bead coating with something like HIV antigen, okay? In the patient sample, we would be looking for HIV antibodies. This is just an example. We do it for many other things as well. If the patient has HIV antibodies present in their serum, and we add HIV like antigens that are coated with a latex particle, we mix them together, we're going to get agglutination. On the other hand, if we have antibodies attached to latex particles, as you can see here, we could look for antigens in the patient sample. Some of the examples that they used were rheumatoid arthritis. Another one that I used was for staph, is we can check for those antigens. Either way, we're going to end up with agglutination in the end. So either the patient sample has antibody or the patient sample has antigen. Agglutination inhibition. This is based on competition between particulate and soluble antigens for limited antibody combining sites. Lack of agglutination is a positive reaction, so that gets people confused. Know that with agglutination inhibition, if we don't have agglutination, it's positive. So this is totally opposite of what you think. This is something that we use to detect antibodies to certain viruses such as rubella, mumps, measles, influenza, parainfluenza, hepatitis B, herpes, respiratory syncytial virus, which is RSV, and adenovirus. Here's an example. This one is agglutination inhibition. So on the top example, we have antigen in the patient sample. We added a reagent antibody to the sample. The antigen and antibody combines together. After that, we add antigen-coated particles. There was no agglutination that took place, so therefore it's a positive test. On the bottom, there's no antigen. We added antibodies. There was nothing for the antibodies to bind to, so we added like a reagent which has antigen-coated particles, and we did get agglutination because it didn't agglutinate with anything in the blood. Positive agglutination equals a negative test. So this is completely opposite of regular agglutination. So here's hemagglutination inhibition. It's very similar, but we throw some red cells in the mix. So the top one, you can see there is antibody in the patient's sample. If we add a viral antigen, let's say this patient on top has HIV. We add a viral antigen. If that patient has HIV, that viral antigen will bind with the antibodies, and then we'll have the red cells added, and if it's negative, you would see a positive test. So there's no agglutination here. On the bottom, we have antigen. There's no antibody. We have red cells, and if they do agglutinate, it's a negative test. So those antigens that are from the test will actually agglutinate with those red cells. That's hemagglutination inhibition. So here's some definitions that can help you when making note cards, Quizlets, whatever it is that you decide to do, going over what we just talked about. So nice little chart form to make it easy and concise. So how do we test for these? Well, one of the newer ways that people have been using, or laboratories have been using, is particle counting immunoassay, or PACIA. This uses nephelometry, and it involves a laser and an optical particle counter to measure the number of residual non-agglutinating particles in a specimen. It measures serum proteins, therapeutic drugs, tumor markers, and viral antigens. Something that's a little bit newer. And lastly, quality control and quality assurance. We want to avoid cross-reactivity by using monoclonal antibodies directed against a very specific antigenic determinant that is unique to the particular antigen. We have to store reagents properly, check the expiration dates, account for sensitivity and specificity, and be aware that just because you have a negative result, it does not rule out the presence of a disease or antigen. There's lots of things that can happen in this type of testing, whether it be having too much antibody, too much antigen, too little antibody, too little antigen. Those can cause a test to give you a false negative. All right, that completes our lecture for today.